So yesterday we have dealt about the pharmacology. Yesterday, especially the cholinergic receptors. We have dealt about the cholinergic receptor synthesis, storage, transmission, and destruction of the cholinergic impulse. So today we will discuss about the pharmacology of the receptors, the continuation of the adrenergic receptors. So in the adrenergic receptors, as usual, you know that the in case of alpha beta receptors, beta 1, beta 2, and beta 3 type of the receptors are there, and the G protein coupled receptor is most of the beta 1, beta 2, beta 3 or beta G stimulatory type of the proteins are there, and usually the beta 1 type of the receptors are activated by adenylyl cyclase and the calcium channels also. The beta 2 is also adenylyl cyclase, cyclic AMP and calcium channel. That's the molecular pathway. Whereas the beta 3 type of the receptors are also coupled with G proteins with stimulatory type of the proteins. So all these three receptors beta 1, beta 2, beta 3 are excitatory in action, whereas the alpha subtypes, alpha 1 type of the receptors are combined with that of the GQ type of the receptors, GQ, sometimes GI or GO type of the receptors, and usually these are combined with varieties of the enzymes, especially the phospholipase C, phospholipase D, phospholipase A2, and also the calcium channels, whereas the alpha receptors do have alpha 2 type of the receptor, alpha 1, alpha 2 type of the receptors. Usually they are associated with especially the inhibitory type of the proteins, this adenylyl cyclase, the potassium channels, and also sometimes the calcium channels, whereas many times the phospholipase C and phospholipase A2. Usually the presynaptic alpha 2 type of the receptors are inhibitory in nature. And the rank of the potency of the agonists. So many agonists are there, namely isoproterenol, epinephrine, etc. So the order of the protein, the order of the efficacy of these receptors is the isoproterenol is more effective than epinephrine, and it is equipotent or little more effective than that of the norepinephrine for beta adrenergic receptor, or else the isoproterenol is having stronger affinity to beta adrenergic receptors, whereas the epinephrine, compared to coming to the alpha receptors, the epinephrine is having more what you call as the efficacy towards affinity towards the adrenergic receptors compared to the norepinephrine, or norepinephrine is more, still more what you call as the potent than that of the alpha adrenergic receptor when compared to the isoproterenol. Whereas in case of beta adrenergic receptor, isoproterenol is highly effective, whereas in case of the alpha adrenergic receptor, epinephrine is more and more selective and effective. And the alpha adrenergic receptor blocker, you have to remember one blocker or the antagonist to and agonist to each receptor. That's always very much required. You can mug up that particular thing. Please prepare a chart and write what is a specific agonist and specific antagonist to a particular type of the receptor. The selective beta adrenergic blocker is propranolol. Beta adrenergic blocker is propranolol. So all these drugs are used in cardiac disorders. Beta adrenergic receptors later are subdivided into beta 1, example those in the myocardium. In most of the cardiac tissue, it is myocardium, whereas the beta 2 is present in smooth muscle and most other side, whereas the beta 3 is present in the adipose tissues and also certain part of the central nervous system. So this beta 3 receptor is about 10-fold more sensitive to norepinephrine than the epinephrine, whereas it is resistant to blockade by antagonism such as the propranolol, very similar to that of the other receptor, especially beta 1, etc. It may mediate the response atypical pharmacological characteristic in adipose tissues, especially the beta 3 type of the receptors are very much involved in lipolysis process, especially in fight and flight reflex, or if the patient is or the human being or the animal is having starvation, then this receptor will be pharmacologically it is active. However, the role of this receptor in regulating the lipolysis is still now unknown, even though it is very well known that the beta 3 type of the receptors are involved in lipolysis process. The exact mechanism how it is going to do the lipolysis, it's once again a question mark. And alpha adrenergic agonists profoundly inhibit the release of norepinephrine from the neurons. Means usually the alpha adrenergic receptors with alpha 2 type of the receptors and with presynaptic in its location, it is going to inhibit the release of norepinephrine from the neurons. Alpha adrenergic antagonists increase the release of norepinephrine, especially acting on alpha 2 type of the receptor, and these presynaptic receptors are designated as alpha 2, whereas the postsynaptic are excitatory, whereas presynaptic are inhibitory in nature. So the alpha receptors which are excitatory are alpha 1, alpha 2 is inhibitory. That's very well known. Several times uh, we are mentioning the same thing. Clonidine is a agonist at alpha 2 than alpha 1 receptor. Clonidine is a specific agonist of alpha 2 type of the receptors, whereas the phenylephrine and methoxamine are selective postsynaptic alpha 1 receptor agonists, phenylephrine and methoxamine, whereas the clonidine is an agonist at alpha 2 type of the receptor, whereas few alpha 1 adrenergic receptors function presynaptically in the autonomic nervous system. We have generalized that alpha 2 is presynaptic and the alpha 1 is postsynaptic, but that doesn't mean that it's a universal truth. Few alpha 1 adrenergic receptors function presynaptically in the autonomic nervous system. That's also possible. In few places, alpha 2 adrenergic receptor are also present at post-junctional or non-junctional sites in several tissue. So that doesn't mean that what we have made a general statement that most of the alpha 2 type of the receptors are pre-junctional, whereas the most of the alpha 1 type of the receptors are post-junctional. This is a general statement that it doesn't strictly mean that all the alpha 2 adrenergic receptors should be presynaptic or prejunctional. So there are so many exceptions are there for the location of this receptor. It also applies to all types of the receptors, including beta 1, beta 2, beta 3 type of the receptors. Here there is a flow diagram how these alpha adrenergic receptors are classified. These adrenergic receptors. So alpha adrenergic receptors and beta adrenergic receptors. So beta is classified as beta 1, beta 2, beta 3. And nowadays also it is told that beta 4 receptors are there and they are located in the central nervous system. And alpha receptors are subclassified as alpha 1 and alpha 2. And alpha 1 receptors are classified as alpha 1A, alpha 1B and alpha 1D. And whereas the alpha 2 type of the receptors are also classified as 4 subtypes: alpha 2A, 2B, 2C and 2D. These are the classification of the receptors, especially alpha and beta. It's very simple to remember. Now, so it's not a difficult thing to remember, especially the types of the receptors. So there is a problem. Please hold on. Yeah, there was a technical problem. Not an issue. It is uh, what you call as. Yes. Here, the subclassification of the receptors already you have learnt, and once again the second messengers which are involved in the what you call as the various subtypes of the alpha. These receptors are phospholipase C, inositol triphosphate. This alpha 1 is excitatory. All this diacylglycerol and calcium. They are all the second messengers or the effectors. Whereas the alpha 2, it is the decrease in the activity of the cyclic AMP. The beta 1 type of the receptors do associated with the second messengers as increase in the cyclic AMP activity. All these three, beta 1, beta 2, beta 3 are associated with increased in cyclic AMP activity, whereas the selective agonists. So please remember this particular selective agonists and selective antagonists is very very important. Alpha 1 selective agonists are phenylephrine and methoxamine. These are the two classical examples. Apart from this also so many are there. Whereas the selective antagonists are prazosin and doxazosin. These are the selective alpha 1 antagonists, whereas the selective alpha 2 agonists are clonidine and clenbuterol, whereas the yohimbine and edoxazine. These are the alpha 2 adrenergic antagonists, whereas the dobutamine is a very specific beta 1 receptor agonist, whereas the atenolol or metoprolol both are beta 1 specific adrenergic antagonists, whereas the beta 2, the adrenergic agonists, are the salbutamol, terbutaline, salmeterol, and formoterol, because the beta 2 is very specifically present on the windpipe or trachea. So, whenever there is a requirement of antitussive agents and anti-asthma agents, all these drugs are used: salbutamol, terbutaline, whereas the opposite action or the selective antagonists of the beta 2 type of the receptors are butoxamine, whereas beta 3 still it is under development, and BRL 37344 is the classical example. Of course, the antagonist is yet to be known. And effects are what happens whenever we activate the particular receptors. See, tissue and effects like alpha 1 type of the receptors. Alpha 1 is always excitatory and postsynaptic in nature. So what happens is the blood vessels are going to constrict, whereas the bronchi it's also going to constrict because alpha 1 is excitatory in nature. GI tract relax. It is inhibitory in case of the because the subtypes alpha 1, 2, D, A, etc. there relax. Whereas the GI sphincters contract. Uterus it is going to contract. Whereas the alpha 2 is usually inhibitory in nature, whereas the blood vessels it is going to constrict, whereas the GI tract it is going to relax. So summation of the action is alpha 1 plus alpha 2. And beta 1 of course it is present in the heart, so most of the time beta 1 is excitatory in case of the heart. In different places like the His fiber, Purkinje fibers, AV node, SA node, and myometrium, etc. The beta receptors are present and they are always excitatory. They are going to contract, means the heart rate will be increased. Beta 2 is going to dilate the blood vessels and bronchi. It's very specifically, we can say that the beta 2 type of the receptors are, their population is very high in case of the bronchi. Beta 3, as usual, it is present in the adipose tissue and also in CNS, whereas its action, how it is going to bring the action, especially the lipolysis, is yet to be understood. In the bladder, alpha 1 causes contraction. Most of the smooth muscles, the alpha 1, except in the GI tract, it is relaxes. And whereas the beta 2, it's going to relax the seminal tract. And heart, most of the time, the very specifically, it is going to increase the heart rate because beta 1 is very specific, whereas beta 2 is also present, but it's inhibitory in nature. So, these anatomically pre-junctional alpha 2 and post-junctional alpha 1 type of the receptors are there. Pre-junctional alpha 2 and post-junctional alpha 1 adrenergic receptors are usually its anatomical location, but there are recent evidences that there is additional heterogeneity of alpha 1 and alpha 2 adrenergic receptors because see the alpha 1 adrenergic receptors and many of the smooth muscle they are going to contract, whereas in case of gastric smooth muscle it is going to relax. So because this is the subpopulation, the distribution of the subpopulation of the receptor. So there are three alpha 1 adrenergic receptor as we have already known: alpha 1A, alpha 1B, and alpha 1D. And of course, in alpha 1, A, B, D, C is not there. Please remember, in case of alpha 1A, alpha 1B, and alpha 1D. Whereas in case of the alpha 2, there are once again four classifications are there: alpha 2A, 2B, 2C, and alpha 2D is also recently found. In the brain, the alpha 2A subtype represents the presynaptic autoreceptor. So coming to the molecular basis of adrenergic receptor function, how at molecular level it is going to act, and activation of all types of the adrenergic receptor, especially the alpha 1 and alpha 2, beta 1 and beta 2 and beta 3 result from G protein mediated effects with second messenger activation with activity of the ion channels. So what is the basis for the action is this system, especially the activation of the receptor involves three interacting proteins. Number one is the first, the receptor, then it's a G protein, effector enzymes. All these are the proteins. The number one is the what you call as the receptor need to be binded with the neurotransmitter, maybe adrenal, especially in case of the adrenergic system, adrenaline or noradrenaline or adrenaline, epinephrine or norepinephrine. So it is need to bind with the receptor, then G proteins will be either activated or it is inhibited depending upon the type of the tissue, and effector enzymes or ion channels are activated. Ultimately, the action is initiated. And beta adrenergic receptor, three type of the beta adrenergic receptor share approximately 60% amino acid sequence. So there is a common amino acid sequence between beta 1, beta 2 and beta 3, whereas the beta 2 adrenergic receptor that interact with each of functional groups on catecholamine against molecule have been identified. There are once again so many type of the beta receptors are available. Coming to the little, not let us not go into detail, but a little structure activity relationships. So the regions appear to create the ligand binding pocket analogies to that formed by the membrane spanning regions of the rhodopsin, a protein, to accumulate the covalently attached chromophore retinal with molecular models of the catecholamine. So there is a ligand binding packet on the particular receptors, and these receptors do have a very specific protein. Whereas the beta adrenergic receptors, the three beta adrenergic receptors share approximately 60% amino acid sequence. Already it is dealt within the presumed membrane spanning domains. The ligand binding packet it has got, and based on the results of site directed mutagenesis, individual amino acids in the beta 2 adrenergic receptor, they are going to interact with each of functional groups on catecholamine agonist molecule have been, these are identified because of the advanced techniques. Yes, thus the protonated amino group of the ligand appears to form a salt bridge between the acidic amino acid, that is aspartate 113, in the third transaminase spanning up of the domain. The ASP reduce is converted in all biological amines receptor. The example, biological amine means adrenergic, dopaminergic, muscarinic, cholinergic, and serotonergic receptors. So this is a very common residue. So interestingly, the rhodopsin or retinal is covalently attached to the lysine 296 in the helix 7, but presumed counter ion for the skiff ways provided by the side chain of GLU113 located on helical turn above the position of this ASP aspartate in the beta receptor in the helix 3. This is about the structure activity relationship. What is the structure of the adrenaline, and then how it's going to fit with that of the receptor, different type of the receptors, or the residues with transmembrane depth. All beta adrenergic receptors do have the relation with that of the adrenalyl cyclase, whereas the all beta adrenergic receptors stimulate the adrenalyl cyclates by in via interaction with G stimulatory protein. Then in addition, G stimulatory proteins can enhance directly the activation of voltage sensitive calcium channels in the plasma membrane of skeletal and cardiac muscles, and this action may provide an additional means of regulating the function of these tissues, especially the certain tissues where excitatory action is initiated. Whereas the cyclic AMP dependent protein kinase or PKC is considered major intracellular receptor for cyclic AMP, and the cyclic AMP dependent protein kinase generally is considered the major intracellular receptor for cyclic AMP, and all the sequential actions are initiated by binding of the adrenaline or norepinephrine to the specific type of the receptors, maybe activation of G protein, then there is incoming flow of the calcium, and also adenosine monophosphate is converted to adenosine triphosphate, then cyclic AMP, and so many cascade of events it is going to take place. The cyclic AMP dependent PKC is generally it is what having its an major intracellular receptor for cyclic AMP binding of cyclic AMP causes dissociation of regulatory subunits as a result of 10,000 to 1 lakh fold decrease in affinity of R or C with resultant activation of the catalytic subunit. This is about the binding of the adrenaline or norepinephrine and subsequent events taking place, especially there is a change or the configuration of the receptor, and the phospholipid is activation of hepatic glycogen phosphorylase. This is an enzyme, hepatic glycogen phosphorylase. So the enzyme promotes the rate limiting step in glycogenolysis, the conversion of glycogen to glucose 1 phosphate. So the example is activation of hepatic glycogen activate this phosphorylase enzyme promotes the conversion of glycogen to 1 glucose 1 phosphate, and ultimately glucose is formed. So this activation itself is a result of cascade of reactions, and especially the cyclic AMP dependent PKC catalyzes the phosphorylation of PKC, thereby activating it phosphokinase and then phosphorylates and activates the phosphorylase enzyme. And this sequence of successive phosphorylations permits considerable amplification of the initial signal. And because of this thing, only a few receptors need to be stimulated to activate a large number of the phosphorylase molecule in a very brief period. See, we are understanding that all the receptors need not be activated by the norepinephrine itself. Instead, only few of the receptors may also be activated by a specific transmitter, and there is initiation of the post-junctional activity. And concurrent with the activation of hepatic glycogen phosphorylase, cyclic AMP mediated protein kinase also catalyzes the phosphorylation and inactivation of another enzyme, that is the glycogen synthase, which catalyzes the transfer of glycosylates from UDP glucose to glycogen. So here concurrent stimulation of the hepatic glycogen phosphate and also the glycogen synthase. Ultimately, these are going to catabolize the body store of the especially glucose from liver and glucose 1 phosphatase. This effect decreases the net rate of synthesis of glycogen from glucose. So especially in case of glycogenolysis process, whenever there is a fight or flight respect is there, then lot of pool of the glucose is released by all the above said mechanisms. The dual effect of cyclic AMP to enhance conversion of this glycogen to glucose and to decrease the synthesis of glycogen from glucose summit the increase the output of glucose from liver. So whenever there is a glucose requirement, what are all the processes, what are all the storehouses? The number one storehouse is the glucose, followed by the muscle glucose, then by the adipose tissue glucose. And similar type of the reaction result in activation of triglyceride lipase in adipose tissues. The another depot of the glucose is adipose tissues, and adipose tissues there will be lipolysis and resultant increased release of free fatty acids. And the lipase is activated when it is phosphorylated by the cyclic AMP dependent protein kinase C. So these mechanisms are little bit what you call as exhaustive and complicated, but you should know how the glucose is produced when there is a fight and flight reflex to combat the body requirement of the glucose. And catecholamines provide an increased supply of the substrate for oxidative metabolism by the mechanism. So heart is an organ which need to act continuously, especially what happens, the stimulation of beta 1 type of the adrenergic receptors leads to positive inotropic and chronotropic action. Means what happens, positive inotropic and chronotropic means the cardiac rate and force of contraction both are going to increase. The increased intracellular concentration of cyclic AMP and enhanced phosphorylation of proteins such as troponin and phospholamban are detected after beta-adrenergic stimulation. So the specific proteins like troponin and phospholamban are detected, especially in different tissues. And the cyclic AMP is an important thing in case of the beta-adrenergic receptors. So all these phosphorylation process are ultimately disposition of the cellular calcium, and there will be intracellular events are going to take place, and positive inotropic response and positive chronotropic response, and both of these are sensitive to calcium channels by G stimulatory protein. See, you may be surprising that the same alpha and beta type of the receptors are existing in so many tissues, but the actions are different. This is because the distribution of the subtype of the receptors in different tissues. The alpha adrenergic receptors, there are 6 distinct type of the receptors especially alpha receptor genes have been cloned, and 3 at alpha 1 and 3 at alpha 2 genes. Already we know that alpha 1A, alpha 1B and alpha 1D, whereas alpha 2 ABCD. There are another three type of the genes, and nowadays alpha 4D are also there, and all the deciduous amino acid sequences conform to well established paradigm of 7 membrane spanning G protein. So the G protein is 7 membrane spanning, while not as though investigated with the beta-adrenergic receptors, the general structure, features, and the relation to the functions of ligand binding and G protein activation appear to agree with those set forth above for the beta-adrenergic receptors. And it is already the action of the alpha adrenergic receptor do vary to great extent when compared to the beta adrenergic receptor, because the relation between the function and ligand binding, both of these are interrelated. So the membrane spanning domains, three alpha receptors, alpha 1 A, 1D, 1 alpha 1A, B and D, three type of the subtype of the alpha 1 receptors, whereas the three type of the alpha 2 adrenergic receptors are no more similar. They are not at all similar, and A and B subtypes, alpha and beta subtypes, approximately 30 to 40%, and variety of the effector cells are distributed throughout the body. And inhibition of a ultra-adrenergic receptors. What happens? The alpha-adrenergic receptor coupled on adenylyl cyclase activity was the earliest effect observed, but in some system, the adenylyl cyclase activity is actually stimulated by alpha-2 receptor, either by G inhibitory BG subunits or by weak direct actions on the G stimulatory proteins. And the physiological significance of these later, especially processes, is currently not clear. Of course, the alpha-2 adrenergic receptor activate G protein gated potassium channel, resulting in membrane hyperpolarization. So whenever the activation of potassium channel, it is a voltage gated process. Usually it is also having the pump, because the concentration gradient outside is less when compared to the inside, and the potassium ions are thrown out and the hyperpolarization status is maintained. So what happens because of this process? There is a dephosphorylation and resensitization of the receptors, and downregulation means the receptor population decreases to longer term desensitization of receptor function, and undoubtedly is mediated by several distinct processes, and this include changes in the rate of receptor turnover, receptor gene transcription, and receptor of mRNA turnover. And these processes are very much complex and poorly understood at present. So this is enough for our knowledge. That means the overall the structure activity relationship is the G proteins, G inhibitory, G stimulatory, and GQ and so many other proteins do have a different structures. However, the what you call as the neurotransmitters, also this norepinephrine and epinephrine, they have a unique structure, but they will bind with the varied structure of the receptors and elicit different type of the activity. And what is the relationship between the nervous system and the enteric and the especially endocrine system? Especially the nervous system and endocrine system have got direct relationships, because the humors means the chemicals are secreted at certain sites to act elsewhere in the body can be traced to Aristotle. It's a very old concept that the humor, neurohumoral transmission, means humors are involved in the nervous transmission. That's why in modern terms, the theory of neurohumoral transmission, by its very distinguishing, implies at least the superficial resemblance between the nerves and the endocrine system. So there is a very clear-cut what you call as the relationship between the endocrine system and nervous system. Already it's very well established. So it is clear that the similarities extend considerably deeper, particularly with respect of autonomic nervous system. So the relationship between the voluntary nervous system is less, whereas the relationship between the autonomic nervous system and endocrine system do have a great relationship. And in the regulation of the homeostasis, the autonomic nervous system is responsible for rapid adjustments to change the total environment. It affects us both at ganglionic synapses and postganglionic terminals by the liberation of chemical agents that act transiently at their immediate sites of release. So the homeostasis is the one of the thing called as the resting state of any what you call as a system, and the autonomic nervous stimulation causes rapid adjustments to the situation. Suppose a fight and flight reflex, the person has to adjust very fastly. Then this is possible because the both the ganglionic synapses and postganglionic terminals do liberate the what you call as the neurotransmitter, and they are stored in the storage vesicles. The endocrine system regulates slower, more generalized adaptations by releasing the hormones into systemic circulation to act distinct, widespread site over the period of minutes to hours and days. Here, the endocrine system is little bit different. For example, the oxytocin acts very fastly on the uterus, whereas the growth hormone takes long time to act. And the half-life of each hormone is totally different from one to each other. Whereas the most of the time the neurotransmission occurs within a very fast what you call as the track, and the central representations of the both system, autonomic system and endocrine systems, are connected with the central nervous system with that through that of the hypothalamus, where they are integrated with each other, and with subcortical, cortical and spinal influences. Different areas of the brain are what you call as controlling the activity of the endocrine secretion and the relationships. The neurohumoral theory thus may be said to provide a unitary concept of functioning of the nervous and endocrine systems. So in which the differences largely relate to distance over which the release mediators travel. So here the earlier it was thought that the neurotransmitter is released in the central nervous system, it will travel throughout the nerves and it is going to do the action. But nowadays it's very clear that the neurotransmitters are stored already in the presynaptic vesicles, and whenever there is a change in the electrical what you call as electrical voltage change, then they are going to be increased intracellular calcium, then the activation of the docking complex, then there is fusion of the vesicles, ultimately exocytosis occurs, and this happens within a fraction of milliseconds. So this is the recent concept, whereas the earlier concept was not like that. And few of the pharmacological consideration. Our interest, yes, we have understood different steps, how a nerve impulse is generated, how it is going to be transmitted, that's the axonal conduction. Axonal conduction, then this is junctional transmission. All these processes are there in the normal physiological condition. And being the pharmacologist, how we can interfere with this process to get the deserved effect, deserved pharmacological effect. That's why in this section presents a classification of drugs that act on the peripheral nervous system and its effector organs at some stage of the neurotransmission. So here yesterday we will recall this because these steps of synthesis of the norepinephrine and epinephrine, the dopa to dopamine, are very important always. Hence we will learn them very carefully. So tyrosine, you know the structure of this tyrosine, and to tyrosine Phenylalanine in the liver is converted to tyrosine with the help of the what you call as hydroxylase enzyme. Dopamine, beta hydroxylase, and tyrosine with tyrosine hydroxylase